So yesterday we talked about the Hardy Weinberg principle. And when we were doing that, we talked about Hardy Weinberg equilibrium, which are all of the things that need to be met, the criteria that needs to be met in order for a gene pool to remain constant. So today what we're going to be looking at is changes in the gene pool, which are kind of just looking at all of the different parts of the Hardy Weinberg equilibrium criteria, but from the opposite side. So why is it that all of those different criteria, if not met, will cause changes in the gene pool? And we'll be going more in depth into that than what we touched on in yesterday's lesson. Okay, so like I said, this lesson is about changes in the gene pool. We're going to be going over things like gene flow, genetic drift, non-random mating, natural selection, and mutations, and looking specifically how they cause changes in the gene pool. You do need to know the different types of genetic drift and their effect on gene frequencies. You also need to know the different types of sexual selection and describe how natural selection works. So some of this will be a bit of review from Bio 20 stuff. So gene pool, just to remind you of the definition, is all of the alleles of all of the genes present in a population. In populations that are not at Hardy Weinberg equilibrium, their gene pools will change from generation to generation. And these are the 5 main causes of those changes to the gene pool. So gene flow, genetic drift, non-random mating, natural selection, and mutations. So gene flow we talked about in yesterday's lesson as well, this idea that gene flow is the movement of alleles from one population to another, also called gene migration. Essentially, anytime individuals come into the population through immigration, they will be bringing in new alleles with them. And anytime there's emigration, so anytime individuals leave the population, they will take their alleles with them. Also, anytime there's breeding between 2 different populations, the offspring will be breeding some alleles with them that didn't already exist in one of the populations because either their mom or dad isn't from that population. So usually gene flow results in the 2 populations gaining more similar alleles over time, so the populations become less different from one another, less distinct, because the alleles are being shared between the 2 populations essentially. So gene flow works against speciation. In order for speciation to happen, if you remember back to Bio 20, you needed the kind of new population to split off from the original population and be separated in some way. And so if you don't have that separation, if instead you have this movement of genes between the 2 populations, then speciation can't happen. As long as they are sharing those alleles, any new mutations that pop up, any new adaptations that pop up will come up in both populations. And so one isn't going to turn into a different species from the other. So this is just kind of a little diagram of what gene flow looks like. So initially, the 2 populations will be very different from one another. If there is no gene flow between them, they're going to have different genes and different alleles. And then as soon as you allow gene flow, allow that movement of individuals, you're going to have a mixing of the alleles. You're going to have both alleles now present in both populations. Until the populations end up just becoming like one giant population because they're so similar. In order to stop gene flow and stop the amount of changes in the gene pool, reproductive isolation is needed. So reproductive isolation is when the 2 populations or the 2 different areas of the population become unable to exchange genetic information. So there's no migration between the 2. There's no interbreeding between the 2. They're completely cut off from sharing any genes with each other. Once reproductive isolation is achieved, then the gene flow will stop, which means that there's not going to be any changes to the gene pool because of gene flow. Obviously, some of the other factors could still apply. But if you have reproductive isolation, the populations will become more distinct over time, which could lead to speciation, where a new species comes from a population. So there are two main categories of reproductive isolation. The first is pre-zygotic isolation, and then the second is post-zygotic isolation. So pre-zygotic isolation is when there is something that prevents gametes from mixing between the individuals of different populations. So pre-zygotic means that there's something happening before a zygote would form to prevent a zygote from forming. And then post-zygotic means that mating happens, fertilization happens, but sometime after that zygote is formed, something is happening to make the offspring either non-viable or non-fertile. So these are the 5 different types of pre-zygotic isolation. Again, that's the one that happens before a zygote is able to be formed. So the first is habitat, meaning that there is some sort of physical environmental barrier between the populations, like a river or a mountain. Think of like your allopatric speciation. There is like a geographical barrier separating the populations, which means they're not able to mate with each other. The second is temporal, where there is a time difference between when the two populations would reproduce. This would be like your sympatric speciation, where it's in the same spot, but at a different time. So maybe the two different populations have different like mating seasons, or one is out in the day and one is out at the night, that sort of thing. Number 3 is sexual behavior, where the mating rituals of the different populations are too different from one another to attract mates. So maybe it's like birds that sing or dance to try and attract a mate, and the singing and dancing of one population is very unattractive to individuals of the other population. And then mechanical isolation is when the male and female genitals are just incompatible, so mating won't occur. Essentially what that means is like the penis of males in one population will not fit in the vaginas of females from the other population. And then gametic isolation is when the gametes will not attract one another or won't be able to fertilize one another. This is usually true in like aquatic populations, where there will be like eggs like floating around or sitting on the lake bed, and then a male fish will just come and like his sperm everywhere. In order for fertilization to happen between those different fish, the sperm has to be like chemically attracted to the eggs in order to find and fertilize the eggs. If you don't have that chemical attraction there, then fertilization won't happen. And then there's 3 types of post-zygotic isolation. There is zygote mortality, which is when the egg is fertilized, so a zygote forms, but then the zygote won't develop from there. The zygote just dies before undergoing like cleavage and becoming a morula or anything like that. Number 2 is hybrid non-viability, which means that the zygote will develop into an embryo and maybe into a fetus, and maybe even into a baby, but won't survive after that. So either the embryo dies or the fetus dies or the baby dies before reaching maturity. Something about that hybrid baby that's half one population, half another, is fatal. And then the third one is hybrid sterility. This is when the zygote develops, so fertilization happens, the offspring is born, it survives, it reaches maturity, but it can't produce offspring of its own. So the hybrid offspring that was produced is infertile or sterile. So that's like what happens when you mate like a horse and a donkey and you get a mule. That is a sterile hybrid that is formed. So those are the types of reproductive isolation that would prevent gene flow. Next, we're going to look at something called genetic drift, which is when the allele frequency in a population changes due to random chance. So it's not changing because you have migration or interbreeding, it's changing because of something completely random. This tends to occur in small populations. This is why yesterday we talked about the fact that in order to be at Hardy Weinberg equilibrium, you need a large population. So random chance doesn't play as big of a role in changing the gene frequencies. If you have a really small population, things that are just completely random can have huge effects on the gene frequencies. Usually it increases the percentage of homozygous individuals, which reduces genetic diversity. There are two main types of genetic drift, the founder effect and the population bottleneck. Genetic drift can also occur just through like really random chance of like the flipping the coin type thing, and maybe like, you know, you have two brown-eyed parents and yet all 10 of their children have blue eyes. It would be unlikely if we do our Punnett square, they shouldn't have all blue-eyed kids. Only like 25% of their kids could be blue-eyed, but it could happen. And so in a small population, that one family that had like a randomly large number of blue-eyed kids is going to really screw with the allele frequency. But the founder effect is a genetic drift that happens when a small number of individuals leave the original population and go become their own population. That new population won't have as many alleles as the original, and it'll be just kind of like random which alleles make it into the new population. So if you look at this example up here, the original population has like 3 different phenotypes of butterfly. And it might be that you had all 3 of those colors try and make it over to the island, and just because of like random bad luck, the white one got blown away or eaten by a seagull or something. And so none of the white alleles made it onto the new island, and therefore that new population now is missing an allele and has less genetic variation. So that's the founder effect. It's this idea that when you just randomly pick a small group from the original population to make into a new population, you might just randomly miss some of the alleles. It would be like if you had a bag of marbles and there was like red, white, and blue marbles, and you just reach in and grab a handful, you might randomly end up with no white marbles in your handful, just because of random chance. That's like the founder effect. Then you have the population bottleneck, which is also called the bottleneck effect. This is when you have some sort of drastic event that is going to wipe out a large chunk of your population. And so this could be something like a natural disaster, like a volcanic eruption or a tsunami. It could be a weather change that causes mass starvation. It might be a new disease or parasite. It might be that like humans came in and bulldozed your habitat. Something really huge happened and most of your population didn't survive. But it's random who in your population didn't survive. It was just like pure good luck and nothing that you did if you survived. Like if an asteroid just like slammed into your population and it just so happened that like 3 of you made it out alive. Usually this is seen when a species is driven to the brink of extinction but doesn't actually go extinct, so there is just a few survivors. And then because the surviving population is so much smaller than the original population, there's a good chance that you have lost some of the alleles from the original population, and it's random which of those alleles didn't make it and which of them did, but the alleles have changed and you now have less variation. Obviously, if they have less variation, they do have an increased risk of extinction because they are less adaptable. And it can also lead to something that's called inbreeding depression. So inbreeding is when species that are really closely related mate with each other. if you have such a tiny population left, all of the species in that population, or sorry, all of the individuals in that population that are mating with each other are going to be really closely related. Even after like a couple of generations, they're all going to be really closely related. They're all going to have really similar genes. And inbreeding depression is when having those really similar genes, because you're all really closely related, leads to more recessive diseases and disorders showing up in the population. So the more closely related individuals are when they mate, the more likely they are to both be carriers for a recessive disease or disorder, which means they're more likely to have a child with that disease or disorder. This is why it's like illegal for siblings to mate with each other, or in most places cousins or anything like that. I mean, aside from the fact that it's gross and creepy, it would also increase the risk of mutations and diseases and disorders and stuff like that in their offspring. In wild populations, it certainly happens. But yeah, this inbreeding depression is when you start seeing those recessive diseases and disorders become more and more common because you don't have enough dominant alleles to kind of cancel them out. Usually, even if the population does bounce back in size, it cannot recover from the loss of genetic variation. There definitely are species that we have seen this in. So cheetahs, for example, today, all of the cheetahs that exist in the world today are all super closely related. Like if you did like, you know, a DNA sequencing, gel electrophoresis of those guys, they would all be like siblings closely related. They have very, very low genetic diversity. And that's because cheetahs survived a mass extinction in the past, that only a couple of them survived, and then all of today's cheetahs are descendants of those few survivors. This also happened with the northern elephant seal, where they were overhunted by humans and went through a bottleneck because of that. Antibiotic resistant bacteria and that kind of stuff is actually also because of bottleneck effects. Essentially, if you like take antibiotics to treat an infection and you don't take them long enough, you'll like wipe out like 99% of the bacteria in your body, and the remaining 1% will be resistant to the antibiotics because they've been exposed to them, and yet you didn't take them long enough to actually kill them off. Which, as a side note, is why you're supposed to take all of your antibiotics, even if you're feeling better partway through taking all of them. Finish what you were given. But anyway, that's how we get antibiotic-resistant bacteria, is that essentially the bacteria that survive have much less genetic variation. And in their case, that means that all of the alleles that are left are resistant to antibiotics. Okay, the next thing that causes changes in gene pools is non-random mating, which is often the case in populations. Usually there are certain traits that will make either the males or the females more attractive to the opposite sex. And then those individuals will mate more and pass on their genes more. This often occurs with something called sexual dimorphism, which is a difference in either appearance or behavior between males and females. So if you can't tell the males and females apart in your species, then there's no sexual dimorphism, and in that case, mating will probably be random. So there's a lot of species of spiders that will actually just like, they can't tell the males and females apart. Even like the spiders themselves can't tell the males and females apart. So if they bump into another spider, they will try and mate with it, regardless if it's a male or a female and whether they're a male or a female. Literally anything they bump into that vaguely resembles themselves, they will try and mate with. So that is completely random, but most species do have some sort of sexual dimorphism where you can tell the males and females apart, or at least they can. And with that usually comes non-random mating. Sexual selection, we've talked about already as well. That is when certain traits increase an individual's reproductive success, which means those traits become selected for and will be more common in the next generation. There are two different types of sexual selection. You will have sexual selection anytime there's competition or access to mates. Usually males are competing for access to females. There is intrasexual selection, which is when individuals of the same sex will try and fight or intimidate each other to gain access to the opposite sex. So usually this is like males trying to become the alpha male and like fight off the other males in order to have access to the females. And then whatever trait makes you better at fighting off the other males, that trait will become more common in the next generation because most of your offspring will have it and you will have more offspring than anyone else. And then intersexual selection is when individuals of the same sex have to attract the opposite sex. So this is usually males trying to attract females either through their appearance, like really brightly colored feathers, for instance, or their behavior, like dancing or singing for birds, or courting rituals, like creating like bowers and stuff like that, which is also something that birds do. And any trait that is considered more attractive to the females will become more common in the next generation because the males with that trait will have more children. Oopsies. Natural selection, we also talked about quite a bit in Bio 20. It is the process where traits that increase survival or reproductive success become more common, and then other traits become less common. Natural selection is the driving force behind evolution. Any individuals that have traits that increase their survival are more likely to mate and pass on those traits, which is why those traits then become more common. Charles Darwin was the one who first coined the term natural selection. He also called it survival of the fittest. The environment will determine which traits are beneficial or favorable. Traits can be beneficial in some environments and unfavorable in others. So like, for example, being really fluffy is great if you're up north, not so great if you're near the equator. In humans, there's actually a disease called sickle cell anemia, which in most places is highly unfavorable because it's painful and can be deadly. However, people who are heterozygous for sickle cell anemia are protected against malaria for some reason. And so the rates of sickle cell anemia are much higher in places that have high death rates from malaria, because sickle cell anemia is actually selected for if it protects you from malaria, selected for everywhere else. There are three different types of natural selection. There is stabilizing selection, where the extremes of a trait are selected against and the intermediate is selected for. So that's like baby birth weights. Too small, the baby dies. Too big, there's complications for the mother might die. So a medium-sized baby is selected for. Directional selection is when one extreme is selected against and the other is selected for, so the trait is driven in one direction and becomes more extreme over time. So that might be like, for instance, if having long tail feathers is beneficial in birds, over time tail feathers will just become longer and longer and longer as long is selected for and short is selected against. And then disruptive selection is when the extreme versions of a trait are both selected for and the intermediate is selected against. So you end up with this kind of like two-hump system. I don't know if I have pictures. Yes, I do. So this first graph here would be the stabilizing selection. This would be the disruptive selection. And then, yeah, this last one is the directional selection. So the dotted line shows the original population, and then the blue is the new population. This last one, this disruptive selection, can result in speciation if it continues. And then the last way to change the gene pool is with mutations. So mutations, like we talked about in the genetics unit, are random changes in DNA. Many are harmful, and if they are, they will be selected against and therefore become less common in the population and might be lost altogether. Every now and then there will be a beneficial mutation. Those will be selected for and become more common over time. And any mutation that stays in the gene pool will increase the genetic diversity of the population. Okay, that is it for today. Let me know if you have any questions.